Hi, I'm Natalie. I'm from the Njoo group, and today I will be presenting two projects. One is on a scalable formal synthesis of plus Etomoxir without pyrophoric agents, enabled by a one-class tandem aldyl Luch sequence. And the other is the results of a project studying the structural editing of gay-ring diversifiable andrographolide analogs for potent activity in breast cancer and colon cancers. So here is a brief outline of what I will be covering. Since it's two projects, it's mainly broken up into two sections. The first of which is covering our attempts to create a more optimized formal synthesis of Etomoxir. So that'll go over the CPT system, what Etomoxir inhibits, as well as our project goals, our current efforts, and in our synthesis and computational modeling, and then our future efforts. And on the andrographolide side, we will be going over our work on A-ring analogs of andrographolide, the rationale behind our project and why andrographolide specifically was selected, as well as the evaluation of the C19 analogs we synthesized. So what is the CPT system? The CPT system is a system of 3 transmembrane mitochondrial enzymes that regulate fatty acid oxidation within cells. This metabolic pathway has major implications in a number of diseases such as diabetes, COPD, asthma, and cancer, thus making it of great pharmaceutical importance, as well as in terms of attempting to regulate this system. In the past, inhibitors such as DHPB, oxyvexylene, or parahexylene, or teglacard have been synthesized and tested in order as potential inhibitors of CPT-1, the main rate-limiting enzyme of the system. However, in past clinical trials, parahexylene and teglacard both failed FDA clinical trials due to a lack of selectivity between two of the isoforms of CPT-1. CPT-1A and CPT-1B, which I will further discuss in later slides. Etomoxir is one of the most well-known inhibitors of CPT-1, and it functions through 3 major structural motifs that it has. The first of which is its arrow ring in blue, which allows it to pi stack to a key histidine residue within the binding pocket. The second of which is its carbon chain, which allows it to have hydrophobic properties similar to the long-chain fatty acids that CPT-1 would usually catalyze. And then finally is its alkylating group, which allows it to irreversibly bind to a cysteine residue within CPT-1. Here is how Etomoxir binds to the cysteine residue within the enzyme, and here is in green is how it is shown non-covalently bonded. Again, I will go over in later slides why attempting to model this reaction in silico is still a challenge today. So Etomoxir did go through phase 2 trials, and it did show initially good promise due to its irreversible inhibition of CPT-1A, its ability ability to treat venicular hypertrophy prevention with by treating CPT-1B as well. However, its lack of selectivity in between the two isoforms, CPT-1A and CPT-1B, what caused its eventual downfall and removal from FDA phase 2 clinical trials. In this clinical trial, it was being used to treat potentially heart disease, which required it to inhibit CPT-1B. However, due to its non-selectivity in between CPT-1A and CPT-1B, it did end up inhibiting CPT-1A, which is the liver isoform. Thus, some of the patients ended up getting induced hepatotoxicity, and thus the clinical trial was canceled due to these poor pharmacokinetic properties. However, previous literature has also shown that a 2,4-dinitrophenol analog of Etomoxir was synthesized and was demonstrated to have lower toxicity and higher selectivity for CPT-1A over CPT-1B. Thus, the switching of the arrow ring as a potential handle for creating more selective analogs is the main focus of this project. We propose that by synthesizing a library of Etomoxir analogs with different aryl fragments varying in hydrophobicity, and like electronegativity and et cetera, will allow us to form new novel Etomoxir analogs that will be more selective in between the various isoforms. In order to obtain these analogs, we decided to focus our efforts in synthesis towards synthesizing this key intermediate, intermediate 1.5. In a previous synthesis in ArcVoc 2004, the 2,4-dinitrophenol analog at compound 1.7 was synthesized in a 6-step synthesis with approximately a 22% overall yield. And though this route did initially show promise due to its use of intermediate 1.5, which would then allow for us to have greater diversification beyond that point towards our own analogs, this route did have one major amongst a couple of other steps that would be unfeasible for a high school student. Most importantly, this step in blue is a homologation using TMED methyl alcohol, but most dangerously N-butylene. This route takes between 16 to 18 hours, depending on scale, which is again unfeasible for a high school student who does not work 24 hours as a researcher. And it also uses N-butylene, which is an extremely dangerous base. It is a pyrophoric that will ignite spontaneously in air and moisture, and thus it is unadvised to scale up this reaction to the large scale that we would want as for a second step in a multi-step synthesis. And moreover, its yield does not merit for this step to be used, does not excuse the danger of this step, because it's only around 66%. So beyond being small scale, you only get 66% of your product. So I'm gonna go a little more into depth as to what like pyrophorics are. So when you use a pyrophoric, you have to use needles. You have to use syringes and like needles with a very high gauge in order to make sure that you don't have so that way you have the lowest possible chance of getting unnecessary air or moisture in the needle, which could then cause your pyrophoric to detonate. So whenever removing a pyrophoric, one has to use a very long needle in order to ensure that when then the syringe is tilted back upwards, you can then fill the top of the syringe with about the amount of nitrogen gas or some sort of inert gas, which is approximately the same volume as the pyrophoric that you've removed. And then that acts as sort of a stopper to prevent any sort of, again, air or moisture from going to your pyrophoric material, causing it to ignite. Then when you add the pyrophoric reagent back to your reaction flask, then you insert the needle and then you tilt the syringe back upwards. But usually, again, that is extremely dangerous if, for example, if your needle cracks or if your syringe breaks or in some way. So this is the arrow pushing mechanism for how our homologation works. It works via a lithiate dianion addition, where first methyl alcohol in a backflush glass is added. Two equivalences of N-butylene are then added, which deprotonates alcohol as well as one of the carbons. And then there is a sort of shared pi system in between carbons 2, 3, and 4, which then it acts as a radical, which then forms single electron transfer towards intermediate 1.1. From then on, the bromine leaves, and this key step over here is the lithiate anion addition, which then forms the bond in between the final products, carbon 2 and 3, and upon acidic workup results in the intended product. So in our efforts to avoid using NBOLE, we proposed an alternative synthetic route that hinges largely on this step in blue. This is an aldol condensation, which this one in particular using formaldehyde, prolidine, and p-toluic acid, followed by a subsequent reduction with sodium borohydride and cerium chloride, then allows us to form the alpha-beta exoalkene motif without the use of N-butylene. In addition, this reaction only takes around 2 hours at most, and it, again, completely removes the use of N-butylene, thus allowing for greater scalability, as all of these reagents are largely safe at room temperature, in air, in moisture, and at large scale. So this reaction works mostly, is all catalyzed entirely by pyrrolidine. So the pyrrolidine bonds with the initial ligand in order to form an imidium complex, which then through a series of protonations and deprotonations from the conjugate base, then gets to the stage where the pyrrolidine bonded to the formaldehyde itself, forming another imidium ligand, which then bonds to the initial substrate, and the initial substrate's imidium ligand. And after, again, another series of deprotonations and protonations from the conjugate base, then results in the pyrrolidine leaving, and the water in the pyrrolidine that the formaldehyde is actually packaged in, then forces the remaining pyrrolidine to leave. And upon the addition of sodium borohydride and cerium chloride, then results in the intended alpha-beta exoalkene. So beyond the previous route, we wanted to optimize this route further. Our old route added in our proposed alternative to NBOLE was safer and better for scalability. However, the epoxidation steps afterwards still suggested the use, this epoxidation and oxidation steps afterwards still suggested the use of reagents such as titanium-4 and ruthenium-3, which are still not recommended for any sort of large-scale synthesis. Thus, we took it a step further by combining our aldol condensation with an asymmetric conjugate epoxidation using a prolinol catalyst on the bottom, 2.3, which would then allow us to reduce the step count one step further from our previous alternative route, as well as avoid the titanium-4 that would be used in the epoxidation. And additionally, by not doing the reduction with sodium borohydride afterwards, we could then do a much more scalable oxidation afterwards, which would then result in 1.4 at a more reasonable yield without the use of ruthenium-3 as well. So this reaction works based off of the previous reaction. These prolinol catalysts can catalyze the aldol condensation similarly to how prolidine can. But however, when it reaches this step over here, instead of doing the reduction, we then leave it with the prolinol catalyst, and where after the addition of hydrogen peroxide, then results in the R epoxide being favored as the major product. And once again, after protonation and hydrolysis, then results in our favored epoxide as the intended product. So now we'll be moving on to our computational modeling side of CPT-1. So in order to figure out which analogs we wish to synthesize for our library, we decided to use virtual modeling in order to screen which ligands, which arrow rings would be preferred for selectivity in between the analogs. The graph on the left is currently our initial results for induced fit glides for our induced fit with CPT-1A in blue and CPT-1B in orange on the graph. As there is no major difference in between CPT-1A and CPT-1B, as most of the results are still within the margin of error. However, we still do believe that some of these ligands may be promising in the future, though we are still rerunning these results in duplicate in order to confirm them. So for CPT-1, our current future plans are to optimize our formal pout, as the first 4 steps, 3 to 4 steps, depending on which synthesis, are not optimized right now. And one major boon to our synthesis would be its higher yields. We are currently attempting to prove its scalability in that regard as well. So we're currently attempting to optimize the first 4 steps. And then secondly is, as I said earlier, finishing the duplicate runs on Schrodinger for our induced fit docking. And then finally would be test after synthesizing all of our analogs to test them with colorimetric assays like DTB and MTT in order to test their efficacy of CPT-1 inhibition and anti-proliferative activity. So now on to the second section of my presentation, which is on Andrographolide. So natural products have been used in medicinal chemistry for a long time. Previous compounds such as Taxol, erythromycin, ivermectin, and others have all been directly from or derived from natural products. For example, but most importantly, some key pharmaceuticals that have been used today have not been just directly from natural products, but they've also been modified, synthetically modified with the use of organic chemistry in order to optimize them for human use. For example, in a key change in between natural products and their synthetically modified analogs happens to be the changing of certain motifs in order to optimize them for greater bioavailability in the body, as well as greater selectivity towards certain targets. For example, the replacement of this hydrogen for a fluoride in fluorocortisone allows for these compounds to have a greater bioavailability and a longer half-life. So one such natural product is Andrographolide, a labdane diterpenoid isolated from the herb Andrographis paniculata. Similar to Etomoxir, it also has 3 main structural motifs that drive its selectivity, reactivity, and accessibility. When Andrographolide binds to its target protein, it mostly reacts through a Michael addition, bonding a cysteine to Andrographolide, which is why the D ring is what drives its reactivity. In a previous work by some of our alumni from the Njoo lab, like Alice, Warren, Celine, and Jesselyn, they showed via a series of C-ring Andrographolide analogs that the C-ring was what drove the reactivity of Andrographolide. And this is in a paper that they published in the Journal of Emerging Investigators in 2002. So along these lines, our lab wished to further explore the potential structural changes that we could do to Andrographolide in order to affect its selectivity or its reactivity. So our next topic of interest was the A ring. And previously, in 2 different papers, there were 2 different proposed mechanisms for Andrographolide from modification of the A ring. So in 2018 in CIREP, it was shown they proposed that a trityl analog would then allow andrographolide to go through, andrographolide would acted through the WNT signaling pathway. However, through a different paper in 2015 in the Journal of Natural Products, they claimed that this acted through the andrographolide itself without any sort of solid ethers on C19, resulted in andrographolide acting through an FKB. Thus, we wish to synthesize analogs that would experiment with the selectivity in between these different pathways. This is a little bit of our results from our experimental study. So we synthesized a series of 8 andrographolide analogs, as well as we tested andrographolide itself in a series of biological assays in order to test its mechanism of action. So we ran MTTs. Our main ones were 96 and 24-hour, and more surprisingly, they showed in 24-hour MTT to have much higher selectivity, the colon cancer cells specifically, over breast cancer cells, which was interesting to note, considering that breast cancer and colon cancer cells it has been proposed in previous literature that breast cancer and colon cancer cells act through have different reliances on beta-catenin, one of the proposed pathways. Additionally, we also performed a SEEP assay, which tracks the level of which we proposed tracks the level of beta-catenin through SEEP, which is a visible colorimetric way to sort of track it. And some of our analogs did have significant, it appeared to show significant inhibition, though there was no clear trend or confirmation of this trend. And then 2 of our later assays were that we co-administered our analogs with a GS3KB inhibitor, CHR99021, and and though there may have been somewhat promising results, these results have been called into question. Though it does seem to suggest that our analogs may act through GS3K beta. And then finally, we attempted to isolate the localization of beta-catenin through immunofluorescence imaging assays. Once again, these results have not been very clear. We suspect that it is potentially due to andrographolide's multifaceted multifaceted properties, being anti-cancer, antibacterial, as it has been deemed basically anti-everything, and thus its potential interactions in a number of different pathways may have not been very clear through our current biological assays. Though with all of these results, we have submitted this paper to Bioorganic Medchem Letters, and as Edward said, we as of this morning, it has been accepted pending revisions. So if you guys want, there's the QR codes for our paper if you're interested. But I suppose this is the end, and I would like to, I'll probably come back to the slide right after, but I just wanted to do this first. So for both projects, I would like to thank Tiffany, Karina, Rushika, and Eric for their work in andrographolide, and then Flora and Rushika for their work in CPT-1 as my fellow lab members in these projects, as well as our collaborators for andrographolide, Dr. Zhang, Dr. Pazzi, Mr. Yamamoto, Dr. Wang, and Dr. Chen, who is also our collaborator for CPT-1 as well. And then we would like to also thank our collaborators in the Zhang lab for their work on MTT assays. but I will end it on this. And if you have any questions, I guess now is a good time.